what the word aneuploidy means?
No, just an abnormal number.
Yeah, it's an incorrect number of chromosomes. So it's someone who does not have, how many are you supposed to have? 46. 23 pairs, which is a total of 46, 23 from mom, 23 from dad. So aneuploidy is whenever there's an abnormal number of chromosomes. And we just talked about that real briefly at the end of class, I think on Wednesday last week. Down syndrome is an example of an aneuploidy, of a condition that's caused by an aneuploidy. They have an extra, it's called trisomy 21. Tri means 3, so they have 3 copies of chromosome 21 instead of just 2. So they have a total of 47 chromosomes in every cell. Down syndrome is one of the few aneuploidies that involves the first 22 pairs of chromosomes, where someone can live a pretty regular length of life with a decent quality of life. There are a few other aneuploidies. I think there's like a trisomy 15 and a trisomy 18, where sometimes those babies can be born, but they usually have very severe problems with development, and they usually do not survive past a couple years old. So those conditions are very rare. So human beings don't tolerate aneuploidies very well. We need to have 46 chromosomes to develop normally. We have to have 2 copies of each one. The place where we seem to have the most flexibility in terms of chromosome number has to do with the X and Y chromosome. And so we're going to focus on those a little bit more today. And some of these are ideas that you saw in the interactive that you started working on after the quiz on Thursday about sex verification in athletes, and kind of the different things that they look at and how that has played into women's and men's sporting events and things like that. That's kind of an ongoing developing thing that is still being discussed in different sports and probably will continue to be for a long time because it's not something that's as black and white as people would like it to be. So we're going to talk a little bit about some of the ideas that you saw in there first. So we talked briefly about the fact that the 23rd pair of chromosomes, if you are biologically female, is XX, and they are homologous. And if you're biologically male, is XY. We talked about that before to look mainly at inheritance in pedigrees and Punnett squares and things like that. Today we're going to talk a little bit more about how that makes someone male or female and kind of how that plays out in the genetics part. So if you're male, you have an X chromosome and a Y chromosome. On the Y chromosome, there is this gene called the SRY gene that is kind of one of the first triggers during the development of an embryo that triggers the developing embryo to have its reproductive organs develop into testes. Embryos start developing reproductive organs kind of before this gene is turned on. But at the beginning, they're just kind of this blob of cells, right, that haven't differentiated specifically into ovaries or testes. And it's the SRY gene that triggers them to develop into testes. So SRY stands for sex determining region of Y. It's part of the Y chromosome that triggers this step in development. Obviously, there's a lot more to developing as a biological male or female than just that one step. There are lots of genes involved in this, both during embryonic development, those early stages, and again, genes that get turned on during puberty that cause the development of the secondary sex characteristics that are all part of this process. So that's one of the first kind of key determining points, but it's definitely not the only gene involved in this. It's just one that you hear about quite a bit in terms of this gender differentiation. Since females have 2 X chromosomes, and the SRY gene is obviously located on the Y, they do not have that gene. And so in the absence of that trigger to become testes, their reproductive organs develop as ovaries, and they become biologically female. Okay? Any questions about that part? An interesting thing happens as development of females progresses, though, and that's a process called X inactivation. Females have 2 X chromosomes in every single cell, But human beings don't actually need 2 X chromosomes in every single cell because males only have 1, right? So you only need to have 1 copy of each of those genes on the X chromosome. And so this process happens where 1 of the X chromosomes in every single cell in a female's body, except for just a couple, is turned off. It's the whole entire chromosome is inactivated, and it condenses into this little structure called a bar body that's visible under a microscope. And that can have some interesting effects. So the reason this happens is so that all human beings in every single cell, except for a couple, in females, have 1 active X chromosome that's coding for traits in that cell. So it kind of maintains the same dose of genetic information in males and females, but the pattern of inactivation is random. In other words, if you're female, you got 1 X chromosome from your mom and 1 from your dad, right? You have 2 copies of every gene that's carried on the X chromosome. They may or may not be identical. Like in this example, this type of cat, what is this, a tortoiseshell cat? Is that the word for that coloring? This cat inherited a gene for orange fur color on 1 of its X chromosomes and a gene for black fur color on another 1 of its chromosomes. In some of its cells, the orange one's turned off, and in some of its cells, the black one is turned off. And so the reason that it has splotchy coloring like that is because of this random inactivation of the X chromosome. There are other examples of that as well. I don't know any off the top of my head. It's possible. This type of pattern, though, it depends what cows you're picturing. And I don't know enough about cow genetics to know for sure if this happens in cows. The thing is, here you're not often going to get like nice, well-defined like spots in certain shapes. That's probably something else, because this would be a lot more random because it's happening in the individual cells of the individual hairs. Does that make sense? So you end up with more like the hairs are more intermixed. But if you look at each individual one, it'll be orange or black.
Some have like hair colors, like some people have like strawberry blonde hair.
Hair color is more complicated than that because there are a lot of genes that contribute to it. I don't know if there are any X chromosome genes that contribute to hair color in humans. If there are, that could be part of it, but that's not the only gene. Like there are genes on other chromosomes too that affect that. The way that this happens, it includes modifications of those histone proteins that the DNA is wrapped around, and methylation. Remember, methyl groups, adding methyl groups is what turns off genes. We talked about that a little with epigenetics a while ago. And so here basically the entire chromosome is methylated and just wrapped up really tight and turned off. This process can be reversed in certain cells in females. For example, the cells in a female's ovaries that are going to produce egg cells. Obviously, if a cell is going to divide and go through meiosis to produce gametes, you want every single egg cell that's produced to have a working 23rd chromosome, right? So in those cells, these bar bodies do uncondense and those chromosomes become activated again so that every gamete has a working copy of the X chromosome. Does that make sense? So turn and talk for a minute. I said at the beginning here that aneuploidies, so abnormal numbers of chromosomes, are a little easier for humans to tolerate when it comes to the X's and Y's. Turn and talk to the person next to you about why you think that might be. So what do you think? Why do you think it doesn't matter so much if you have like an extra Y or if you're missing a Y? Yeah.
Because the X and Y chromosomes really just code for reproductive organs and like mostly normal lives.
That's definitely true with the Y chromosome. So most of the genes on the Y chromosome do have to do with the development of reproductive organisms. And, no offense to the biological males in the room, but the Y chromosome doesn't really matter that much because obviously you can develop as a perfectly normal human without a Y chromosome, right? Obviously it matters to the proliferation of the species, but not to the development of an individual, right? Because half the population survives without one just fine. Why do you think it doesn't matter if we have, if we're missing an X or if we have an extra X? Why doesn't that matter so much?
It's just coding for the same.
And if we have those of us that are biologically female that have two X chromosomes, are both of them working anyway? No, we just said one of them gets turned off anyway. So if you have a couple extras, it doesn't really matter. So humans seem to be a lot more tolerant of aneuploidies here than anywhere else. Yeah. Yes, I think so. I believe that's how that works, because that does exist. That's not the first one on my list. I don't remember if I put that on this list, but I think it was on that website. So these are some examples, and I don't expect you to memorize the characteristics of every single one, but there are actually a lot of variations in the sex chromosomes. And I just put a little bit of notes on here. So people can be XXY, that's called Klinefelter syndrome. This is one of the more common examples you'll hear of. I think it happens a little more frequently than some of the others because it actually has a name, so I think it's a little more common. These people are biologically considered male, but they're sterile. A lot of these people who have aneuploidies in the sex chromosomes have trouble reproducing, which should make sense because if you have 3 chromosomes, it's hard to evenly divide them, right? So you end up with sperm or egg cells with the wrong number of chromosomes in them, which are much less likely to develop. Some of these characteristics have other side effects. Sometimes the other side effects don't show up until puberty, when the secondary sex characteristics don't develop the same way or maybe don't develop at all. And that sometimes people don't even know they have some of these conditions until they reach puberty. Some of them can also have side effects that might cause learning disabilities or affect mental development, intellectual development as well, but not always. So there's a huge variation in how many symptoms or effects this would have. So XYY is also possible. That one's not as common, but that's also possible. Normal sexual development, no specific symptoms, at least as far as we know. Trisomy X, so that's the one that we were just talking about, also occurs. Again, usually healthy, no unusual features, but they are for learning disabilities, and they apparently are able to reproduce. I don't know why that one is tolerated better in terms of reproduction than others, but maybe again because X chromosomes are inactivated. And monosomy X is another one that you should, if there's one of these that you kind of remember, it should probably be this one. It's called Turner syndrome, because this is the only known case where a human being can develop with 45 chromosomes. With a monosomy, where they only have a missing chromosome, nowhere else in the first 22 chromosomes there is no case where someone will even reach it to birth if they're missing an entire chromosome. So this is the only viable monosomy in humans. They're phenotypically female, but they're sterile. I would assume because half of their gametes are not going to have a 23rd chromosome at all. I don't know. There might be some other mechanism that doesn't work when there aren't 2 X chromosomes. It's probably part of the process of triggering those cells in the reproductive organs to go through meiosis and develop gametes. Something hits a stopping point and freaks out because they're missing a chromosome and says, well, we can't do this at all. Even though logically you could say, well, but it might work okay. The cells don't know that. Other questions about that? Okay, so if you didn't finish that interactive, I just stuck this in there kind of as a reminder. 5th hour, we had some College Board issues, and so I did tell it. I don't think I actually officially changed the due date, but I told them if they didn't have it finished by today, that was okay. So if you're still working on it, please get that finished and turned in if you haven't done that one yet. Any questions there? Because then we're going to kind of shift away from talking about the X and Y chromosomes and just go back to genes on all the chromosomes. Okay, how are we doing here? Good. All right, so now our last kind of brand new topic for the genetics unit, which will take us a couple days, at least part of tomorrow, is we're going to talk about linked genes. Important note, we are not talking here about X-linked or Y-linked. Those mean so when we say the phrase is X-linked or Y-linked, what we mean is it is located on that chromosome, right? An X-linked gene just means it's located on the X chromosome. A Y-linked gene means it's located on that chromosome. And so in terms of inheritance, we use the word linked because the inheritance of that trait is linked to the person's biological sex, whether they're male or female, will influence their chances of inheriting certain traits. But now, and that's why I said we're kind of shifting gears, we have to let go of the X and Y. What we're talking about here are linked genes in the sense that they are physically, I think this is probably on here. I don't need to rewrite it. Not on the same gene, I should say on the same chromosome. Okay, I will write it. So linked genes are genes that are physically found together on the same chromosome. They are part of the same long strand of DNA. They're just located in slightly different parts of that strand of DNA. And so because they're on the same chromosome, linked genes do tend to be inherited together. This is the exception to Mendel's rule of independent assortment, where every gene kind of does its own thing and they don't affect each other unless they're physically located on the same chromosome. So in this picture, gene 1 and gene 2 are not linked because they're on completely separate chromosomes, and each chromosome is going to do its own thing during meiosis. And what happens with gene 1 will have no effect on gene 2. Gene 1 and gene 3 are not linked. They're on different chromosomes. But genes 2 and 3 and 4, which you can't really see because it's written in yellow, but 2, 3, and 4 are all located right together on the same stretch of DNA. Those 3 are linked genes. And unless crossing over happens right between, like if crossing over happens right here, and moves that little piece of DNA, that can unlink genes, and we'll get to that a little bit later. But if that doesn't happen, genes 2, 3, and 4 are all going to move. That whole chromosome is going to move into the same gamete, and that offspring will get all 3 of those genes together as a package. Does that idea make sense?
So if there's a gene like on the lower half of that, are they?
They are, but they are much less likely to be inherited together. Like I said, we're going to get back to this idea in a minute, but the further away they are, the more likely it is that crossing over would happen. And like if there's one way at the top and way at the bottom, there's almost 100% chance that crossing over is going to happen somewhere on that chromosome. And so they're going to act as if they're not linked, even though they're technically on the same chromosome. Other chromosome or questions? Any questions about this? Just the definition of what linked genes are. Okay. So you have a diagram in here that shows some fruit flies. And before we really start talking about this in terms of gene linkage, this shows you some experiments that were done when scientists were first beginning to figure out that genes can be inherited together like this, and independent assortment doesn't always hold true. But I want to point out a couple of things on here that's terminology that you've probably heard before and may or may not fully understand what it means. First of all, are you familiar with the P generation, the F1 generation, and this would be considered the F2 generation? Have you heard those terms before? Yeah, you can think of it, so P stands for parental generation. F1 officially stands for first filial. Filial means like son or daughter. It's probably Latin or something. So first filial, you can think of it as the kids. And F2 then is second filial, or like the grandkids. They are words that you will frequently see when you're doing multi-generational crosses like this. It's often done with fruit fly experiments. You will also see these sometimes on pedigrees, the P generation, the F1, the F2. I suppose you could have an F3, but honestly, I don't know if I've ever seen that. The other thing I want you to notice here is the notation that they're using. So if we look at the parental generation here, one of the flies they're calling the wild type. Okay, so wild type is a way to describe genetics of a trait. It's generally the most common in a population, and I think it always means dominant, because I'm not sure how this would work if it didn't. The biggest difference you'll notice is the way that they're written. In this case, the wild type flies are gray with normal wings, and you will notice that the gene for color is not written as a capital letter. It's written with a little plus sign as kind of an exponent. That denotes the wild type. So this notation means that it's the wild type. It basically is like the capital B that we have been using. I do not know why this notation is frequently used, especially with fruit flies and genes like that, but this is how they write it. And so over here you can see this one. They say it's a double mutant. This one is black, so it just has a lowercase b with no plus. So if we were to make a little key over here, for the fly color, the dominant allele is the wild type, which makes a fly gray. The recessive allele doesn't have the plus sign. It's not the wild type, and that makes the fly black. The other trait we're looking at here is wing shape. You can see this one has little tiny, they call them vestigial wings. The word vestige means like a trace, like they're not fully developed. And again, they're using that same kind of notation. So the wings here, they're saying VG plus, that's the dominant trait, that's normal wings. And VG without the plus is recessive, and those are vestigial wings. So those are kind of the two traits we're looking at here. Like I said, later this week, you're going to be doing a virtual fruit fly lab, and so you will see this notation for more traits. And it comes up very frequently when they present you, when they kind of describe an experimental scenario related to genetics and fruit flies especially. You'll see this a lot. So I just want you to pay attention and notice that so that you recognize what that means. So now if we look at the cross that they did, you can see the two parental flies were homozygous for both traits. So one of them was wild type. It had gray color and normal wings, and it had two dominant alleles for both. And they crossed that fly with a double mutant. So it was homozygous recessive for color, and it was black, and homozygous recessive for wings, and it had those vestigial wings. So that means all of the offspring of that first cross, the whole F1 generation, were dihybrids. They were heterozygous for both traits. They had a dominant and recessive allele for both. Then they did a test cross. You remember what a test cross is? Kinda. Look at who they crossed this fly with and turn and talk to the person next to you and see if you remember what a test cross is. We talked about it long, long ago when we started this part of the unit. Okay. Not sure if you remembered it completely or not. A test cross is always when you cross the individual that you're really testing, so we're really experimenting on this one, with one that is homozygous recessive for all of the traits that you're concerned about, in this case for body color and wing shape. The reason we want to cross it with something that's homozygous recessive is we now know the phenotypes of the offspring are completely decided by this parent, right? Because this parent can only give the recessive genes, so that's not really ever going to determine the phenotype. What will determine the phenotype is whether they get the dominant or the recessive from the parent that's heterozygous. So the scientists who did this experiment were trying to test whether the gene for body color and wing shape were linked. And as they thought about this in advance, they said, okay, if these genes are linked, so here is our F1 dihybrid parent, right? This is just the genotype that's over here. I just read it bigger. If this is the genotype of this parent, if these genes are not linked, then they're going to sort independently, which means some of the flies will get this combination, some will get this combination, some will get this combination, some will get all four of those are possible. That's our foil, first outside, inside, last. And they should all occur equally. Does that make sense to everybody? That's just basic meiosis. You'll get all four combinations equally, which means, so these are the four phenotypes that will go with those flies. One of them will be just like this parent. It'll be heterozygous for both traits. It'll be gray with regular wings. One of them will get all the recessive genes. It'll be black with vestigial wings. And then you'll have a couple that are called recombinant phenotypes. The one that's gray with vestigial wings and black with normal wings. So you get all the combinations. And you should get them all in equal proportion. So if these genes are not linked, you should get them in a 1-to-1 to 1-to-1 ratio. We okay with that? If these genes are linked, that would mean that these parental genes, the two dominant genes and the two recessive genes, are on the same chromosome. So then you wouldn't expect to see any of those mixtures, right? They'd either get both the dominant genes or both the recessive genes because that's how they're found on the chromosomes. So if you get linked genes, you should have half this one and half this one and none of these at all. Everybody following the logic so far? So they actually did this cross to see what happened. And they got a ratio of about 5-to-5 to 1-to-1, which means they had about, 12. 5/12 wild type, 5/12 black with vestigial wings, but 1/12 gray with vestigial wings, and 1/12 black with normal wings. First of all, which one does this look closer to? Do you think those genes are linked? Probably. We could do a statistical test to confirm. We'll talk about that in a minute. But it's closer to this, right? You definitely got a lot more of those parental phenotypes, the ones that look like their parents. But you still had a couple that were recombinant. How do you think these happened? Yeah. Remember, we already said crossing over can unlink genes. We're going to talk more about that in a second. Right now. Okay, so crossing over is the way that linked genes can become unattached from each other. That process is called genetic recombination. And we actually already looked at this a little bit. You remember the meiosis in Sordaria with the black and tan spores in those sets of 8 that we looked at? We were actually looking at how often crossing over happened with that color gene. So we already looked at this a little bit in that lab, but we were just looking at one trait, not two of them. So crossing over is the way that these genes can become unlinked. A couple of vocab words that you want to notice on here. When we talk about linked genes and the offspring of those crosses, the parental types are the ones that look phenotypically like the parents. So they have the same genetic combinations that make them appear the same as the parents. So if one of our parents here is yellow and round, here's our offspring that are yellow and round. They're going to look just like the parents. These are homozygous recessive, so they're going to be green and wrinkled. That's the other phenotypes for each of those. This is the other parental phenotype. So each of these, we expect to get some of those and some of those because that's what the parents look like. Recombinant means they get a new combination of traits. That's kind of what that definition says over there. That isn't found in either of the parents. So here we have one that's yellow, like this parent, but it's wrinkled like this parent. So that's a new mixture of traits. And that's the other recombinant. It's round like this one, but it's green like that parent. The more recombinants that we find, the more crossing over has happened. So by calculating the percentage of recombinant offspring, that actually tells us how frequently crossing over happened between these two genes. At least the species that they do these experiments with. Not all of them, but there's tons of different varieties. All right. Last thing for today. So when we actually do these crosses to test whether genes are linked, you can look at the numbers, like when we get that 5 to 5 to 1 to 1 ratio, you can say, yeah, that really looks like those genes are linked. But as with everything in science, using our best judgment to determine whether it's close enough isn't good enough. We need statistics, right? So here's another example where we're going to try and determine whether these genes are linked. So in Cosmos plants, purple stem is dominant to green and short petals is dominant to long petals. If we do a test cross with an F1 plant and a recessive plant, so this is just like what we did. We have a heterozygous, that's our F1, and a recessive plant. Our expected ratio, if these genes are unlinked, again, is 1 to 1 to 1 to 1, because it would be equally likely that offspring would get each of these combinations of genes if they're not linked. Make sense to everybody? So 1 to 1 to 1 to 1 is the ratio. The expected number, this is kind of like what we did in chi-square. To figure out the expected number, we have to add up what our total is here. Let's see, is that easy math? Is it 500 exactly?
9.
4, 6, 8, 9. Yep. So our total there is 900. If we divide that by 4, what's half of 450? 225. So the expected number of each would be 225. Again, this is one of those cases where we look at those numbers and say, pretty close, right? What would you do to figure out if it's actually close enough? Yeah. Chi-square is actually used with genetics more than any other situation that we've looked at this year. We've used it in a couple other situations just to practice, but genetics is where chi-square is really used a lot to try and determine whether the offspring that you get match the ratio that you predict. So chi-square's goodness of fit test. Does the data match closely enough for us to accept it? So here our hypothesis is that these genes are unlinked. In a chi-square, we want to state that a little more like a statistical null hypothesis. We're going to talk a little more about this. But when you're doing a chi-square with genetics, what you're really saying is there will be no statistically significant difference between our observed results, so that's the number we actually saw of each phenotype, and whatever ratio is expected by the type of inheritance that you're testing. So here, we expect our observed results will match the 1 to 1 to 1 to 1 ratio predicted by the inheritance of unlinked genes. One really important difference that you will notice as we start doing chi-square with genetics here is that the expected numbers are not always going to be equal. Because if you think about just a really simple Punnett square where the two parents are heterozygous, you don't expect equal results. You expect 3/4 dominant and 1/4 recessive, right? So your expected comes from whatever your Punnett square prediction is. Then you would go through and actually do your calculation. And I don't know if we'll actually do this today, but it's observed minus expected squared over expected, right? And you add that all up to get your chi-square value. If you would like, you may actually do the math. We're going to be doing more chi-square later. Once you get your chi-square value, what do you have to do with it to figure out what it means? Yeah, you remember that little table that you have on your equation sheet? So first you do this, then you compare it. We call that the critical value. In this case, we have 4 categories. So how many degrees of freedom do we have? 3. It's the number of categories minus 1. So in this case, there's 4 possible phenotypes, 4 categories, 3 degrees of freedom. For this example. If your chi-square, that's the symbol for chi-square, is lower than the critical value, what do? You accept or reject our null hypothesis here. You accept. Because if the number is small, it means the difference is small. That's how you have to think through it. If the number is small, the difference is small, which means there's no difference. The difference is not statistically significant. In this case, if we got a chi-square value lower than the critical value, that means that the little difference we saw was just random chance, which happens, but these genes are pretty close, or these results are pretty close to that 1 to 1 to 1 ratio that shows us that these genes are not linked. If our chi-square was over that critical value, we would reject the null hypothesis, which would mean maybe these genes are linked, and then we need to start looking at that. Or some other complicated genetic thing is going on in terms of how these genes are expressed. Questions? All right. That's all for today. Tomorrow we're going to talk a little more about linked genes and how we can use them to draw maps of where genes are found on chromosomes. Which is kind of fun.
I just have a question on